Adjacent to the stomach are the three major accessory organs that are involved in the process of digestion. So now is as good a time as any for us to talk about them before we go on and see what the small intestine and the large intestine do. Let's talk first about the liver. And the liver, you might be surprised to know, is actually the largest of all of the glands in your body. It is a gland. It secretes lots of different substances and it stores lots of different substances. Anatomically, it's divided out into four lobes. There's a right lobe, there's a left lobe. This is on the anterior or the front portion. On the posterior portion of the liver, there is a caudate lobe. That's this little lobe here that's associated next to the inferior vena cava. And then sitting immediately adjacent to the gallbladder, that's that green structure there is a gallbladder, is this other lobe, which is called the quadrate lobe. So if you're looking anatomically, you're trying to remember which is which, just remember the quadrate lobe is always associated or next to the gallbladder, and you'll be in good shape. So the liver does lots of different things. It is a place where we absorb lots of things from our food. First, it gets absorbed in the small intestine, and then it gets shuttled to the liver, and it's stored there for later use. So things like extra amino acids that are present in the blood, extra glucose in the blood, extra lipids in the blood, vitamins and iron also. These all get absorbed and stored in the liver, and then when such times as the maybe vitamin levels are too low for a particular vitamin, say vitamin A or something in the body, then the liver will release some of that vitamin, stored vitamin, out into the blood to elevate levels back to where they should. The liver is also responsible for detoxification of the blood in part. Now, you'll remember that the kidneys do a major detoxification because they are the point of removal of nitrogenous waste products and some drugs. But even before blood gets to the kidneys, if it hits the liver first, the liver also has some cells that have very active smooth endoplasmic reticula, and these smooth endoplasmic reticula manufacture a host of substances that have the ability to detoxify different toxins. For example, you have an enzyme called alcohol dehydrogenase present in cells in your liver, in the hepatocytes. That's the name for a liver cell. And that alcohol dehydrogenase is an enzyme the hepatocytes make, and it's used to break down ethanol, the substance that's in things like beer and wine and gin and tonics, and it helps to break that down into other less toxic substances. Other things the liver does, we know that it manufactures a whole lot of different proteins, things like albumins and different clotting factors like fibrinogen, for example, and thrombin. It manufactures angiotensinogen, which you'll remember is the inactive version of angiotensin, and that becomes the active version angiotensin when renin is secreted from the kidneys. So you might be wondering to yourself, how in the world is it that the liver can do such an amazing array of duties? And I'm going to show you very briefly here kind of which cells are responsible for these roles, but I want to really highlight first just how critical the liver is to your body's function. If you have liver failure, you're in really dire straits because your ability to manufacture the proteins that are normally found in the blood is compromised. Your ability to store and release lipids, amino acids, glucose, nutrients, vitamins, minerals, that's all compromised. Your ability to detoxify the blood of toxins like alcohol is compromised. So loss of liver function is a serious life-threatening condition. Now, the liver is able to do all of these different duties, and on top of that, it's also able to manufacture bile thanks to the activities of the hepatocytes that are found in the liver. And if we looked up close at how the liver cells are oriented inside the liver, they're not exactly like this, but it kind of gives you an idea. You kind of have these hexagonal groupings of cells, and at each of these intersections here, there is usually something called a hepatic triad, and that consists of something called a bile ductule, a branch of the portal vein, and a branch of the hepatic artery. So at each of these locations you have one of those triads. Okay, and if this were another hexagonal, you'd have the same thing here. And at the center of each of these is a what's called a central vein. Now these hepatocytes, and in this picture here the hepatocytes are represented as these little pinky cells, are the cells that do the manufacture of the proteins, they're the ones that store the nutrients, they're the ones that do the detoxification, and they manufacture bile. And the way that they do this is that when blood enters into the liver, it commonly comes from the hepatic portal vein. The hepatic portal vein is a vein that connects the small intestine with the liver. So we're going from one capillary bed in the small intestines to a second capillary bed in the liver. By definition, that's a portal system because I did not first return to the heart. This is called the hepatic portal vein because it's carrying blood to the liver. So here's my branch of the hepatic portal vein here. Blood enters in through here, and that blood has just come from the small intestine. And if you've eaten a meal, that blood has probably absorbed nutrients from the food that you've digested. Those nutrients, the ones that are in abundance, like let's say you had a heavy protein meal and you've got tons of amino acids now absorbed into the blood, will then be shuttled through past these hepatocytes, and the hepatocytes will store those amino acids for later use. Eventually the blood will make its way to the central vein, and then it will eventually exit out of the liver. Meanwhile, now that's happening at each of these corners, right? You've got a portion of a branch of the hepatic portal vein at each of these corners, moving blood inward. As it moves by, the hepatocytes are picking up nutrients that are in excess and storing them. At the same time, those hepatocytes are also manufacturing bile. Now, the bile that is being made by the hepatocytes gets secreted out of the hepatocytes, and it gets picked up in these bile ductules, which are like little canals for collecting bile. What's weird is that the direction in which the bile moves is opposite from the blood flow. So bile moves outwards towards the edges of the hepatic triad. And this whole thing here is, by the way, is a hepatic lobule. So we're moving out toward the corners of the lobule if we're bile. If we are blood from the hepatic portal vein, we're moving inward towards the central vein. There's also, you'll notice here, a branch of the hepatic artery that's bringing oxygenated blood to the liver. It too travels towards the central vein, and along the way it would be delivering things like necessary oxygen for the hepatocytes so that they could continue to function. There are a number of large canal-like collection areas called sinusoids. You'll remember sinusoids are a type of modified capillary that are kind of irregular shaped, and they're fenestrated. And these are what collect up all of the items that the hepatocytes allow to pass by, or that in times of need that the hepatocytes actually release into the central vein so that they can enter circulation. There's also in and among all these hepatocytes another type of cell called a Kupffer cell, named after a guy named Kupffer. Modern terminology just calls this a hepatic macrophage, and that should give you a darn good idea of what Kupffer cells do. They are macrophages that hang out in the liver. They are surveying the blood as it passes by, particularly the blood that's just come from the small intestine, and they're ensuring that there are no pathogens present or no other toxins present in that fluid. Things like viral particles, fungal spores, etc. So this picture here gives you another viewpoint of that hepatic triad that I was talking about on the previous slide. And you can see here that they've shown you, let's do this way, that these hepatic lobules are sort of somewhat six-sided in shape. And again, at each of the corners here, you have a collection of three structures. You have part of the hepatic portal vein, you have part of the hepatic artery that's bringing oxygenated blood in, and you have what's called a bile canaliculus, a bile duct. Sorry, this is a bile duct here. The canaliculi feed into the duct. So these three together constitute a hepatic triad, and obviously they're very important for keeping the liver connected with the rest of the body so the liver can serve its function of storage and release of nutrients when needed, and for manufacturing bile. So where does that bile go once I've made the bile? And you'll notice that I've skipped over all of the physiological processes involved in the manufacture of bile, because we will touch on it later if we have time. We'll see. But for now, we won't worry too much about it. Okay. When we get to chemical digestion, I will tell you exactly what bile does in terms of its role in helping you get digested food into your intestine. When bile is manufactured in those hepatic lobules, it then feeds into bile canaliculi. Those feed into a bile ductule, and then these ductules eventually begin to merge with one another and feed into even larger bile ducts. Okay, so we have these larger bile ducts, and these all congregate together. These bile ducts then connect up with this organ right here called the gallbladder. So you have a left and a right hepatic, you have the bile ductules go into the left and right hepatic ducts, or bile ductules, they're called both. They feed into what's called the common hepatic duct, or the common bile duct. Okay, and these guys then feed into the gallbladder here. Now, I would never ask you to know, the journey that bile makes from an individual hepatocyte until it gets to the gallbladder. But it would be fair to ask you, what is it that the gallbladder does? And the gallbladder is basically serving as a place to store bile until it's needed. Okay, it's just a storage point. You'll notice that if I erase all of this stuff here, that there is a common passageway here. And that color is not going to work. There's a common passageway here that connects the gallbladder to the small intestine. And then immediately adjacent to it, there's another pathway that also connects the pancreas into that same region. Where those tubes meet up, okay, the tubes that meet up from the gallbladder and from the pancreas, they meet up together at a sphincter on the small intestine wall called the hepatopancreatic sphincter. Very appropriately named because it combines items that are manufactured in the liver with items that have been manufactured in the pancreas and dumps them together right here in this location simultaneously into the small intestine. So as I said, the purpose of the gallbladder is to store bile for us. The liver makes the bile, the gallbladder stores the bile. And bile is really it's a hodgepodge mixture of a bunch of different types of molecules, and it includes a lot of cholesterol. Your liver is able to manufacture about 85% of all the cholesterol you need. So most people think you get most of your cholesterol from a high cholesterol diet. No. You naturally make 85% of all the cholesterol you need. Only about 15% more comes from the diet. Other things in the bile besides cholesterol are some different types of fats, a few different types of minerals, sometimes, for example, calcium. There's some phospholipids in there. There are what are called bile acids and bile pigments. And the bile pigments are important because these are what lend that a kind of a yellowish-greenish color to the bile. And that's because of the presence of a substance called bilirubin. Bilirubin is this yellow-greenish kind of substance, yellowish-green, and it is what lends that particular color to the gallbladder. Later on, we'll learn that in the large intestine, there are bacteria that will convert that bilirubin into something called urobilinogen. That has kind of a brownish color to it, and that's part of why that's a big part of why our feces are brown in color. We'll also learn later that about 80% of all the bile that's made, we use it to do some things with fats that I'll tell you about, but then that bile, it gets actually reabsorbed in the small intestine, and it can be reutilized again on another go around. It goes back to the liver and kind of gets spruced up, if you will. Gets chemically modified to make it functional again, and it goes back into the gallbladder. In essence, though, if you really want to think of this in simple terms, the bile is really composed principally of waste products. Breakdown products and waste products that come from other things. Phospholipids come from the breakdown of cells. The bile pigments are coming from the breakdown of red blood cells when we break down that hemoglobin. So these are waste products principally, and the cholesterol is there to store them. The gallbladder is there to store them. And if I have a sufficient amount of highly concentrated bile and it's allowed to sit in the gallbladder in a super-saturated solution, and this can happen if people are super dehydrated or if they tend to roller coaster through diets where they lose lots of weight and then they gain weight back. They lose lots of weight, they gain weight back. This super-saturated solution can produce gallstones. Gallstones are kind of similar to kidney stones in that they have things like calcium carbonate in them. They have a lot of cholesterol in them, of course, because bile is made of a lot of cholesterol, and there's a lot of bilirubin in them. And of course, gallstones can be very painful to pass, just as kidney stones are very painful to pass. So I think that's about as much as I want to talk, about the gallbladder. Except to say that don't forget that it is just a storage place. It's not a place where we manufacture bile. The liver actually makes the bile. Let's talk about the other major accessory organ that's located near the stomach, and that is the pancreas. And the pancreas is actually, if you were to look where the pancreas is, it's actually situated immediately behind or posterior to the stomach. And I was hoping maybe this picture would show it a little better, but you really can't see it there. So anyway, take my word for it. The pancreas is located posterior to the stomach, and it's right at about the intersection between the upper portion of the small intestine, which is called the duodenum, and where it intersects with the stomach up here. And that's not really a very good stomach, is it? So there's my stomach. Here's my small intestine down here, and right kind of at the intersection back here is this yellowish structure here called the pancreas. And the pancreas is one of those unusual organs in the body in that it has both endocrine and exocrine functions. We already learned the endocrine functions when we looked at the endocrine system, and we learned that the pancreas manufactures insulin from beta cells. It manufactures glucagon from alpha cells, and then there are those delta cells that modulate how much insulin and glucagon get released and when. So we're not going to worry about the endocrine function because we've already learned about that. What I want to focus on is the exocrine function of the pancreas. More of the tissue of the pancreas is devoted to exocrine function than to endocrine function. And the exocrine function of the pancreas is that we have cells in the pancreas who are responsible for manufacturing a whole bunch of different enzymes that help digest starches, they help digest lipids, they help digest nucleotides, they help digest proteins. And these are all cumulatively secreted together into the duodenum of the small intestine, and we call it pancreatic juice. Another thing the pancreas manufactures quite a bit of is bicarbonate ions. And those bicarbonate ions, when they get dumped from the pancreas into the small intestine, help to neutralize the pH of the chyme that's come from the very acidic stomach. And in fact, we add so many bicarbonate ions that it takes the pH from being very acidic into being a very alkaline pH. Well, not very, but somewhat alkaline pH. So here are some examples of the types of enzymes that you find in that pancreatic juice. Pancreatic amylase is an enzyme that degrades or breaks down or hydrolyzes, splits apart, starches, carbohydrates. Okay, amylases all do that. They're involved in carbohydrate breakdown. So we'll learn more about this when we get to chemical digestion. So I'm not going to go into the specifics now. Pancreatic lipase is an enzyme that helps to degrade or to break down lipids, lipase for lipids. So this is for breakdown of fats like triglycerides. The pancreas also manufactures ribonuclease and deoxyribonuclease. I bet you could tell that these are two enzymes whose job it is to break apart RNA and DNA into individual nucleotides. Notice in every one of these instances, we're trying to take big molecules and chemically break apart bonds to make them smaller so that they're more easily absorbed. We have a bunch of enzymes that help to digest proteins, including things like the pancreas makes an inactive form of an enzyme. The inactive form is called trypsinogen. The active form is trypsin. Helps to digest proteins. Chymotrypsinogen also is manufactured. That's an inactive form. It gets converted to chymotrypsin, also involved in protein digestion. Procarboxypeptidase is also a precursor enzyme. And don't ask me why they didn't put the OGEN on the end of it like they do with all other inactive enzymes. This one they called it procarboxypeptidase. The active form is carboxypeptidase. Another enzyme that helps in the digestion of proteins. So these are all made by the pancreas and secreted into the small intestine. The trypsinogen here is the first one to get converted into trypsin, and it's converted into trypsin by another enzyme the pancreas makes called enterokinase. So enterokinase converts trypsinogen to trypsin, then the trypsin converts chymotrypsinogen into chymotrypsin, and it also converts the procarboxypeptidase into carboxypeptidase. Now we have to make sure that we keep an eye on when the pancreas is releasing enzymes into the small intestine. Obviously, it would be really wasteful to just dump them into the small intestine all the time when we don't eat constantly. So we only want to have pancreatic secretions really become dumped into the small intestine, into the duodenum, when it's appropriate, when we have food coming, or chyme, I should say, coming from the stomach and entering into the duodenum. And this is regulated, the secretion from the pancreas is regulated in part by the parasympathetic nervous system, our vagus nerve, that vagal reflex tone. The vagus nerve is what helps to manage when pancreatic secretions are allowed. Sympathetic nervous system would inhibit the release of pancreatic secretions. We also have more localized hormones and paracrines, things like gastrin from the stomach can also help with the timing of when pancreatic secretions are released. CCK, which stands for cholecystokinin, is also another secretion that helps to organize when pancreatic secretions are released. And secretin from the stomach also helps to stimulate when the pancreas releases enzymes. If we take cholecystokinin, CCK here, cholecystokinin is often secreted from the duodenum when you've had a high-fat meal. And it's a way for the duodenum to signal to the pancreas, hey, I just received a bunch of lipids, and you need to boost your secretions into the duodenum here so that I can get these lipids digested fast. Okay, that's enough on the accessory organs. Next up, small intestine.